Good afternoon, everyone. So, seems like crowd is less because of conference or might be Thanksgiving. So we'll continue to discuss on Axon guidance. So last class I talked about different mechanism for Axon guidance. So we touches some of these four mechanism I have discussed here. One is contact mediated repulsion. So if you have negative the signal which axon doesn't like, or it repel, it doesn't go into the direction, so we call this as contact mediated repulsion. Then we talked about some of these example of contact mediated attraction that happens because of interaction between the axon, and you have fasciculation, formation of axon bundle. You can see here. Then we talked about some of these other mechanism, like where you have the diffusing chemicals that can also attract or and repel the axon. So we talked about chemo attraction and chemo repulsion. So we will continue to talk about some of these example of these four categories. So I have listed here one by one. So we'll talk about first some more example for the contact mediated attraction. So the next slide. So here this study was done in a Drosophila model, where they have identified a molecule called as FAS2, fasciculin 2, which the fasciculin 2, which helps in fasciculation of the axon. So this is example of a contact mediated attraction. In that category it falls. So here this study is done in the Drosophila, and this fasciculin 2 is identified through Drosophila genetics. So by doing gene knockout or knockdown approach, they have identified that the fasciculin 2 is important for axon guidance through contact mediated attraction mechanism. So here this is the wild types that they have done 3 sets of experiment. This is the wild type. Then you have the fasciculin 2 loss of function mutant. Then this is the rescue. This is the rescue or the fasciculin 2 gain of function. Gain of function means they have overexpressed fasciculin 2, that they call as gain of function. And they have rescued means they have the mutant. They have overexpressed transgene in this mutant background. They call this as a rescue construct. So we'll go one by one. So this is the wild types. So here you have 2 distinct like signaling pathways operating. One is MP1 signaling pathway, another is VMP2 signaling pathway. So you have the neuron from the MP1 signaling pathway, and then you have the neuron that is through the MP2 signaling, VMP2 signaling pathway. So these 2 neurons interact, MP1 and MP2, and the interaction is facilitated by another the TCC group of neuron. So this you can see that because of the interaction, you have formation of this axon, fasciculation of axon. So this is they have identified this in early stage 14. Now in the loss of function, what you have, when you have loss of function of fasciculin 2, and this interaction that you are seeing here is mediated by fasciculin 2, which is the cell adhesion molecules. So then when you have loss of function of fasciculin 2 or in the mutant, so you can see that in the control you have this nice sheath of this nice bundle of this axon because of the normal fasciculation, because of normal expression of fasciculin 2. But when you have loss of fasciculin 2, we can see that this VMP2 or the MP1, they are not forming the axon, like they are not fasciculating, they are not forming the axon bundle. So then you can see that those axons are being separated here. So those 2 pathway that you are seeing is MP1 pathway or MP2 pathway is not working normally. That's why you have don't have the fasciculation. So this is seen in fasciculin 2 loss of function mutant. But when you overexpress in this mutant background, so this is in mid stage 13. So when you have loss of this fasciculin 2, then you overexpress fasciculin 2 transgene in this mutant background. You can rescue all these things, that defect you are seeing here. You can see that you have normal fasciculation. So you have normal bundling of the axons. So this is through the between the contact between the axon, 2 axons. So that's why you call it as a contact mediated attraction. So that is done by the fasciculin 2. The question might come in the exam. I might put like, what is the function of fasciculin 2? or I might ask, like, I will put few molecules which is required for the exon guidance, something like that. So I might frame question like that in this in this slide. So just to show that kind of what kind of question I will ask. Okay, so so here loss of function means you have a loss of acetylene-2 or little expression. If you do RNA experiment, you can knock down, you can reduce the expression of acetylene-2. So you have little bit acetylene-2, that's why too little. Okay, or you entirely can knock out by using CRISPR-Cas9 technology, you can knock out the gene. So now I'll be talking about, it is not only between these two axons, the interaction can happen between the receptor present in the axon and also in the extracellular matrix. You have the substrate. You can also have the axon can grow nicely depending upon which substrate you use. So here they have used two different substrate. One is polylysine, another is poly-D-lysine, different version of the lysine. And here you can see that the axon are nicely extending their like neurites. So they are nicely growing in the surface. And as I mentioned, that it depends on which type of substrate you are using, and that can also enhance the axon guidance process. So here this is showing that how this is happening through the different substrate. Just I want to highlight these are different substrate, and people have used to show that how substrate actually regulates axon guidance. So they have taken 5 different substrate, like laminin nitrocellulose substrate. These are artificially synthesized, or laminin polylysine substrate, or N-cadherin, or polylysine, or L1 substrate. So so axon guidance always doesn't depend upon the adhesivity. So you can see that the 4-1, like the polylysine, has high adhesivity rate, but the growth, you can see there is very less growing, and the axon are actually growing very less in this substrate, but it's highly adhesive. So depends on, there's a range of this. So you can see that the laminin nitrocellulose adhesivity is less, but the growth rate is more. So here I have I will say the best will be N-cadherin. You can see that it has moderate adhesivity and also growth rate is moderate. So N-cadherin is might be the ideal substrate for for the axon guidance. So you can see that this is something you can remember, N-cadherin. But these are the different substrate people have used to show that how substrate can modulate axon guidance. So that is I talked about contact mediated attraction, because you have axon that can directly contact with other axon, or it can contact with the substrate that you that you have in the lab. So then we'll continue to talk about other like example of contact mediated repulsion. So the axon is growing along the substrate, but suddenly it encountered a substance which inhibiting its growth. Then what it does is it will not grow in that direction. Sometimes the axon will grow in that direction but will stop growing. That is called as contact mediated inhibition. But sometimes what they do is they will not grow into this direction, but they will repel away and grow into other direction. So that we call as contact mediated repulsion. So that is also some of these neuron behave contact mediated repulsion mechanism to grow their, like to guide their axon. So here one example is afferin and 2 signal. So afferin 2 signal is a negative signal, so axon will not grow into that direction. So here no treatment. So this is only the control one. You can see that, and the affectin is stand with the antibody. Sorry, you can use the dye or antibody to stand actin. So you can see that in the control where they have not treated afferin A2, you can see that they had nice projection of filopodia. The growth cone is nicely organized. You can see the actin affectin bundle. This is how the growth cone looks like. But when this axon encountered a negative signal, which is inhibits the which the neuron will not grow into this the signal, where the signal is there, the neuron will not extend its axon. You can see that you have loss of this, this structure, which is shown here in the control. So this is called as example of contact mediated repulsion. So growth cone collapse when you culture with this signal, that is epinephrine A2, chemical like the signal. You can see that the growth cone suddenly collapse. So this is called as contact mediated repulsion. So you can see that another treatment, another signal is the SIMA3A. So here, no treatment. You can see that you have nice organization of the filopodia-like projection in the growth cone. You have growth cone is nicely growing, but when you put this chemical, the SIMA3A, which inhibits the growth of the growth cone, you can see that some of this growth cone, the filopodia-like projection which are present in the control, they are actually retracted. So this is the growth cone collapse in the presence of negative signal, either SIMA3A or in the previous slide, the epinephrine A2. So these are contact mediated repulsion signal. So sometimes, so when the axon encountered the repulsive signal, it will not suddenly stop growing. It takes some time to avoid that signal. It can also step back and can grow in other direction. That is something is highlighted here, like repulsive cues trigger cessation of the growth cone motility for a few minutes. So when it encountered a negative signal, it will stop growing into that direction. It will pause for for some times. It will not extend its axon to this negative signal. Rather, it will kind of come backward and grow in other direction. So here you can this is the figure that's showing how this thing happens. So you can see that this is axon, this is another axon. This is the growth cone is extend kind of trying to contact with this axon. So here this is after 11 minutes, it is growing into the direction. You can see that it is kind of making contact, but after some time when it get the negative signal, like if you treated this sample with the SIMA3A or like epinephrine A, what it does is it will retract all these processes in the growth cone that you have the filopodia-like projection. You can see that these are being retracted, and it is tries to make kind of retracted, or it stop growing into that direction, and it extends its projection in other direction. You can see that it extends here. This is the original where it's supposed to make contact, but now it started extending its filopodia-like projection or in the growth cone to other direction. You can see that it is making contact with other direction. It stopped growing in this direction. So finally you can see that after 64 minutes, it's supposed to make contact here, but it is making contact in other side. So it stops growing into that direction where you have the negative signal. It stops growing, but after some time, what it will do is it will kind of extends its filopodia-like projection in the other direction to make contact. So the growth cone sometimes stops growing and stop kind of wait for some time, and it will extend its filopodia-like projection to the other direction. Okay, so this is called as contact mediated repulsion mechanism. So it repel. So when it found this the negative cues or negative signaling molecules, it stops, it repel. So that's why it is called as contact mediated repulsion mechanism. Okay, so now we'll continue to talk about more example of this. The example 2 is like motor. So how this cues, that the negative cues or positive cues, we are talking about the negative cues, which is called as contact mediated repulsion mechanism. So here another example is contact mediated attraction mechanism. That is when you have this motor neuron, which from the spinal cord, the motor neuron extends its axon and make contact with other, mostly in the muscle. So so here I am talking about connection between the neuron to muscle. So here you can see this is the spinal cord, part of the spinal cord. Here you can see that the axon that you are seeing here, they are extending its projection to the you have something positive cues in the muscle, and that's why the axon is attracted to this positive cues. And first what it does is make kind of they contacted with each other, kind of fasciculate with each other, that is called as axon fasciculation, and they enters into, or they project into the muscle. So this is all the steps are shown here, like the positive signal that is present in the muscle could be either the laminin or a fibronectin in the extracellular matrix. So then when they get the positive signal, what they does is they selectively fasciculate. So you can see that the axon are fasciculating each other, and depending upon the gradient, like if it is a chemoattractant, so here in the case you have the chemoattractant in the muscle, so they innervate to the muscles. So this is how you have the axon is guided from one particular compartment to other particular compartment. Like in the in vivo, I talk about this. This is in the in vivo model. So here motor neuron innervate to the muscle because of you have secretion of this. You have this chemical present in the muscle, like the laminin or fibronectin in the extracellular matrix of the muscle. That's why you have attraction towards these chemicals in the muscles. If some negative signal like ephrine or SIMA3A will be there, then the axon will not be extending its projection to this direction. It will be extending to other direction. So that I talked about in the cell culture experiment, where they have treated this cell with the SIMA3A. The axon is not directing or projecting its axon to that direction. It is making contact with other cells. So these are the example of how the signal actually, either if you talk about in the in vivo model, here in the motor neuron and muscle contact, or in the in vitro model, like in the cell culture experiment with the SIMA3A. So this is how you have extension of axon or projection of axon to the different compartment. Another example is another signal is the ephrine signal we talked about before. So here what happens is you have the neurons that is coming out from the spinal cord. So you can see that these are the neuron that came out from the spinal cord, and they form kind of bundle of the neuron at the ventral root. So this is the ventral root, and the neuron in the ventral root, they project into one particular region, and these are called as peripheral nerves. Okay, so the peripheral nerves also follow certain path. So they do not extend its projection to the region where you have ephrine A signal. So if you remember the previous lecture where I talked about, you have a neural tube formation, then you have either side, you have the somites. In the somites, you have these are the mesodermally derived structure. You have anterior side, your posterior side. The anterior side, the rostral side, you have this is the rostral side of the somite. This is the posterior side or caudal side of the somite. The caudal side of the somite, you have the ephrine A signal, more ephrine A signal, and ephrine A is is a negative signal. Okay, it's a negative signal. So axon will not extend its projection to the ephrine A region. So in the somite, you have the anterior side, you have the posterior side, or the caudal side. So you have ephrine A signal. So the axon which are coming, or the nerves which are coming from the spinal cord, and they actually join together at the ventral root to form these peripheral nerves. The peripheral nerves basically project into this anterior side of the somite. You do not see the projection of this peripheral nerve to the posterior side of the somite because you have ephrine A signal. So that's why you see all these kind of peripheral nerve concentrated in the anterior side of the somite. And this is shown also in the lab. This is the in vivo model, and this is the in vitro. So here they have different lanes for the here in this lane you have the ephrine A signal. You do not have ephrine A here, and then this is all the duplicate or triplicate they have done. So where you have the ephrine A, you do not see the axon is extending to this to this region. So you can see all these axon they are present in the negative when you do not have the ephrine A signals. So the signals basically guides the axon where it should go. So the afferent A is guiding actually, axon. So you should not come to this region because it has afferent A, which is a repulsive signal. Okay. So so more example for the like contact mediated attraction or contact mediated repulsion to define contact mediated attraction or a repulsion mechanism for the axon guidance. So here the experiment was done in the chick embryo spinal cord. What they have done is they have surgically removed some part of this spinal cord and they have displaced into different regions. So they, you have this spinal cord, intact spinal cord. They suppose they surgically remove this part and they put here. What happens to the axon guidance? Or whether the axon will project into the region where it is supposed to project, or they deviate from their original orientation or original projection. So here the setup experiment. So in the control, you have this, the axon coming out from the spinal cord. They are projecting into target muscle. So here these are you can see that the axon is nicely projected to the target muscle. M is muscle. But when they did the small displacement, you can see that it was here, but now there is a space in between. You can see there is no space. That means they have displaced this, neuron that is coming out from the spinal cord to a other region. It's still, projecting its axon because it's not that far. You can see that it's not that far. And they might take some other path. So here you can see the path is kind of very regularly, organized. But when they displaced it, you can see that it's not following the similar path. But when they did a large displacement of this part of the spinal cord to other region, you can see that it's still showing projection to the target muscle because you have these positive cues in the muscle which attract the axon. So you still see the projection, but they are not taking the right path. They are taking the aberrant path to reach to the muscle. Okay. And sometimes what that does is they can also project to the other compartment because it is not placed in the region where it should be. So sometimes it innervates to the wrong muscle. So mostly the thing that I want to highlight here is, you have these cues, positive cues, which can actually guide your axon to reach their target, at least in these two cases. And even in this case also, you still see this projection to the muscle, but it is taking the other, aberrant path to, connect to the muscle actually. Okay. So that is how these cues can actually guide your axon to reach their targets. So not only the chemicals or signals, but also the Hox genes. Hox gene plays an important role. I mentioned several times. It required for the pattern formation. A lot of things happens with the Hox gene. Hox gene can also regulate the guidance of axon. So here, this is the Hox gene cluster. You have different cluster, Hox A, Hox 4, Hox 5, like Hox 10, 11, something like that. So Hox 6, 9, and 10, they are called as columnar Hox. They produce different sets of transcription factor, and interaction of this among the Hox gene has been shown to regulate axon guidance. So here they have looked into how the Hox 6 and Hox 10 interaction helping to guide the axon from to the limb. So here, this is shown in lateral motor column. So you can see that, because of interaction of Hox 6 and Hox 10, the axon is projected to the limb. Or interaction among other Hox genes can also, extend its axon to the limb muscle. So these are Hox 3, 4, 5, 6, 7, 8, and this is shown in the forelimbs. You don't have to remember this which Hox gene is interaction is essential for innervation up to the muscle. So you just remember that Hox gene is also plays an important role for axon guidance. The question I might set like, what are the factors required for the axon guidance? I'll put Hox gene, I'll put the SIMA3A signal, like that I will put. Then you have to choose the correct answer for that. Okay, so you don't have to remember which Hox gene is required for axon guidance in the limb, mostly in the lateral motor column or in the forelimb, something like that. So I am not going to ask that question, such type of questions where you have to remember. Okay, so So this is all about the Hox gene interaction and axon guidance. So here another so this axon guidance is not a simple process. It's a it is regulated by multiple things. It is regulated by the chemicals, signals, or the Hox genes, or the the target muscle. You have the different the signal that is present in the muscle, or it is through the transcription factor, different transcription factor. So here you have so different pools of transcription factor can also regulate axon guidance in response to the muscle cues. So the muscle cues means the transcription factor that is present in the muscles. So you have here they have talked about different sets of transcription factor and innervation of the nerve to different region of this organism, like the muscle. So here they talked about the medial motor columns, how this transcription factor regulate axon guidance. So I'll talk about that. Then they have talked about the lateral motor column, and then they talk about the sympathetic neurons. So we'll go one by one. So we'll talk about first medial motor columns. So these are the transcription factor. It is called as ISI1 or ISI2, LIM3 or LX3. So these are the transcription factor that is actually expressed in the medial motor columns in the target tissue, the target muscles. So here this is the neural tube. So here this is the target muscles, and you have the notochord here, and you have the ganglion here, and you have two different negative signal, which is SIMA3 signal, mostly SIMA3 signal. This is the posterior side, this is the dorsal side, this is the ventral side. So ventral side you have the Ephren A signal, which is a negative signal, and in the dorsal side you have a SIMA3 signal. So definitely the axon will not extend as the axon will not extend to its projection to this region. You can see that it is extending to this to this muscle which have FGF growth factor. Okay, they are not extending its axon to the SIMA3 or Ephren A signals. And this type of kind of mechanism seen in the medial motor column medial motor column muscles. Okay, mostly the axial muscles. And if you look at the sympathetic ganglion, here you have the SIMA3 signal, negative signal. You have the Ephren A signal, and this is here you have the FGF. So now this neuron is projecting its its axon to the ganglion. So depending upon which type of neuron you are talking about, which type of muscle you are talking about. In the axial muscle, you see this kind of mechanism, and in the sympathetic ganglion, you see this kind of mechanism. Okay, so and you have the expression of this transcription factor in this ganglion, like ISI1 ganglion, expression of ISI1 transcription factor. And you have here, you have expression of ISI1, 2, 3, or LX3 for the medial, innervation to the medial motor column. So the thing is that you require transcription factor for the axon guidance, or you require, here in the previous slide, I talked about you require the expression of a Hox gene, you require the different signal for, cues, that is called as cues for axon guidance. Okay. So, the Ephren present in the ventral. So this is the ventral, this is the dorsal, SIMA3 in the dorsal. So that is what I've, shown that, how this, different, signal can guide the axon to different compartment. So here, showing more example, kind of receptor. So SIMA3, you have, SIMA3 receptor for the SIMA3 signal, you have Ephren receptor for the Ephren signal. So here expression of a receptor is also important for axon guidance. So here, here it is showing two sets of neuron in the dorsal axial muscle. So you have two sets of neuron, that like MMC neuron or HMC neuron. So the MMC neuron, they express different sets of transcription like receptor, or HMC neuron express different sets of receptor. So MMC neuron express Ephren AS receptor. Okay, so you require transcription factor, or you require signal, and also the receptor expression can also helps in axon guidance. So here in HMC, mostly in the MMC neuron, you can see that it is innervating to this dorsal axial muscle because you have expression of afferent AS receptor in this neuron. Here in other case, like in the ventral axial muscle, so mostly the HMC neuron, you have expression of SIMA3 and 3F receptor. That is innervating to this ventral axial muscle. So expression of a receptor, expression of signal, those things are required for the axon guidance. So you have, since you have expression of this receptor, SIMA3A, 3F receptor in this, HMC neuron, they are, kind of extending its axon to the ventral axial muscles because ventral axial muscle is specific for SIMA3 and 3F receptor. Similarly, dorsal axial muscle is specific for afferent AS receptor. So here, receptor is also important for axon guidance. More examples here showing that like SIMA3F or afferent BS receptor. Here showing classification of different afferent like afferent AS receptor. Here you have afferent AS receptor, all these things. So you require different receptor, different signal. Hox genes. Lot is a very kind of networking. It's kind of complex things for the axon guidance. You require a lot of things. It's not only one like a mechanism. There are multiple mechanisms that actually operates in different tissue or different compartment. So here ventral axial muscle you have SIMA3F, and in dorsal axial muscle you have the afferent AS. Okay, so that is what I'm showing that which afferent is specific for which receptor. So afferent A is specific for afferent A4 receptor, like that, something SIMA3A for this one. So all these things I have kind of shown that the receptor and its chemo like repellent or chemoattractant. So here more example showing that innervation of this axon to different muscle like ventral limb muscle or dorsal limb muscle. So that is something I kind of highlight here. So you have different neuron, you have MMC neuron, you have LMC neuron. So different neuron can extend its projection depending which type of signal they have in different compartment. So in the ventral limb compartment, you have expression of SIMA3A, and you have the dorsal limb compartment, you have SIMA3F. So and each neuron that you are seeing like LMCI neuron is specific for SIMA3F, so something like that. So depending upon the signal, depending upon this expression of different receptor, you have axon guidance. Okay, so some of these things I have mentioned here, like which neuron is specific for which receptor. Okay, so here again the same thing. Don't have to mug up or don't have to remember anything. So I will ask according like kind of analytical things, like I'll put few signal like which are negative signals, like which negative signal means the axon will not, either if it will extend its axon to this region, it will not grow. That is called as inhibition. It will inhibit its growth. It will retract its philopodia. That is called as axon collapse. So I'll put like SIMA3F, afferent A, and all these things among these four, which are required for the axon collapse, something like that. Okay. So then we'll talk about some more example of this mechanism. Maybe I'll talking about some of these example for the chemoattraction or chemorepulsion. Like these are the diffusible. So whatever I have shown or talked before, these are all contact mediated, either contact mediated attraction or contact mediated repulsion mechanism. You send your axon and contact to the surface. If it is a positive signal, then you will grow. If it's a negative signal, you will retract or you will take different path. So because you need the contact, that's why it is called as contact mediated. And sometimes you have the diffusible, chemicals or attractant or repellent that is also helps in axon guidance. So we'll talk about some of these for the repulsion, chemorepulsion, diffusible molecules, or chemoattraction. So we'll talk about these two, touch on these things. So here the example showing again the both mechanism like contact attraction, chemoattraction, or chemorepulsion. So here talking about formation of commissure. So commissure is a band of nerve tissue connecting the two sides of the spinal cord. So here, this is the midline. Okay, this is one side of the spinal cord. This is another side of the spinal cord. Basically, these two sides is joined by this thick of nervous tissue that is called as commissure. So here, if you see this one, this is the dorsal root ganglion. This is the wild type. So this is the motor neuron, and this is one half the spinal cord. Is the another half the spinal cord. You can see that these two spinal cord half is joined by a band of nervous tissue, and this is called forming commissure. And here one gene is deleted, and this is called netrine gene, which is a positive signal. You can see that you don't have this formation of the commissure. We'll talk about this. So here, what it is shown there, two diffusible kind of chemoattractant or chemorepellent molecules. So here, netrine or slit. Netrine is a positive chemoattractant, and this is a chemoattractant, and slit is negative. Is a repulsive chemorepellent actually. So here they talked about how the axon is extended. It's how the axon is going in response to two signal, like either netrine or slit. And you have the receptor for netrine, which is DCC, and you have the receptor for slit, which is ROBO. Okay, so here this is example of chemoattractant. So this is the two sides of the or two half of this the spinal cord. So in the spinal cord you have the molecule either netrine, which is in red, or slit, which is in blue. So and here this is a growth cone in the containing netrine receptor, and netrine is a positive chemoattractant molecules, diffusible molecules. So when you have diffusible molecules present in this in this spinal cord, you can see that this axon containing the netrine receptor is going to these directions. Okay, so it will be growing to this. So when it grow, it connects these two two sides of this, two half of this spinal cord. That's why you see this type of structure. But when you have a loss of netrine, you can see that you don't connect the two half of this spinal cord. So, and this axon, and because of the axon guidance, you have the connection between the two half of the spinal cords. So then another example is the robo slit interaction. So robo, the axon basically express slit receptor. When you have slit receptor, you have the robo. The robo express the slit chemicals. Robo is a receptor, but slit is the diffusible chemicals. So when the axon find this diffusible chemical, which is slit, present in this spinal cord, it extends its axon, and because you have the robo receptor, so then again you have formation of this commissure-like structure. So the positive signal, the netrine or DCC interaction or slit robo interaction is essential for for formation of this commissure, formation of a joining of these two half of the spinal cords. So either it is a negative or positive. So you require this kind of diffusible chemoattractant or chemorepellent for formation of commissure, and this happens because of the axon guidance. Because axon is guided to this depending upon the signals, either slit signal for the robo or netrine signal for the DCC receptor. Okay, that is what is shown here. Like when you have the netrine signal, the axon is growing to this to this direction. But when you have this robo or slit, you can also see the axon is growing according to the either to the in context of the chemorepulsive molecules or chemoattractive molecule. Like here I have talked about these two mechanisms. They are making this commissure-like structure because of this axon guidance. Okay, so that's what I have shown that. So then there are other mechanism has also been shown where axon can grow depending what type of tissue you are talking about. So here they have talked about in the optic chiasma. So here, this is the part of this thalamus, required for the visual processing. So here you can see that, here you can see that the axon is following a different trajectory pathway. Either it will go to this compartment or to this compartment. So this side of the compartment is called as, this type of movement to this compartment is called as ipsilateral axon goes towards the ipsilateral side or to the contralateral side, this side. So here, what happens is when axon come, kind of following the trajectory path in the optic chiasma, of the brain. So what it does is, here in this point, it will define in which direction it will go, and that is depends on the which type of signal they receive. So here, if they receive, positive signal, which suppose the axon want to innervate to this region, the that is called ipsilateral tract. So you will see that the axon will repel from this region. So you will have expression of certain chemical in this region. So axon will not go in that direction. It will go in that direction. So this type of kind of axon pathfinding or axon guidance is shown in optic chiasma of the brain. So I'll talk more about this in the more detail. The visual, how the axon guidance happens in the visual systems. So here this is shown in the again in the in vitro condition. So here you can see that the growth cone is rapidly extending its axon. This is after 55 seconds, and this 55 minutes. Sorry. And this is you can see that after 90 minutes it's forming nice projection to the visual in the optic chiasma region. But sometimes like here in this slide I talked about, here it will decide where it will go, and whether it will go in that direction or this direction, depending upon the expression of chemicals. So here, if it gets a repulsive signal in this region, so it will not extend its axon to this region. It will extend its axon to this region, this region. This is called as ipsilateral optic tract. So this is something is highlighted here. Like here, after around 65 or 135 minutes or 170 minutes, then when it you can see that after 10 here in this, at least in this condition, the lower half, you can see that here the axon is extending to this direction because it is getting positive signal. But when you get negative signal after 10 minutes, look at this bottom panel, you can see that axon is not extending this direction. It is rather kind of stopping itself. It is growing in the inward direction. So you can see that the axon is projecting its axon kind of to other directions. So this type of movement happens because of expression of certain negative signal, and that is why you have you have projection of neuron different compartment. So this is something shown in the lab here. It get the negative cues here, then the axon is not extending to this direction. It is just stopping its growth and kind of growing in other directions. You can see that it was not extending like this, which is you are seeing here. So this type of growth or axon guidance has been shown in retinal ganglion, retinal ganglions, where you have projection of neuron to the other compartment or other region because of expression of a certain repulsive signal. Okay, so this is something like here. So when it then how the axon is projected into this like contralateral optic tract, because then in this condition what you will have is you have expression of again negative signal in this region. Then the axon will not extend its axon to this direction. It will start extending its axon to this direction. So this happens. This is regulated by the cells. At what time it will express negative signal for one region and positive signal for other region. So it happens in the in the tissue itself. Okay. So just we are touching the last part of this like axon guidance. So this work is done by famous scientist who got Nobel Prize for this. Mostly Roger Sperry, so who was in this lab. So they have given a certain hypothesis for this projection of axon to different compartments. So they have used this model, amphibian models, where they define how expression of certain chemical in one compartment is guiding the axon to that region. So I will talk about that. So this is called as retinotactile projection in amphibians. So I don't want to spend much time on this, what has been done, but I just want to show that what kind of experiment they have done to say that the axon is actually projecting to particular compartment. So here the experiment was done in amphibian's eye. So some of this neuron in the retina of the amphibians is projected to the other region of the brain called as tectum region. So you have this is the retina of the the frog. So you have specific site present in the retina called as nasal site, and another is the temporal site. In the tectum of this amphibian brain, you have anterior side and posterior side. So what happens normally is the axon is projected from the temporal side to the anterior side and nasal side to the posterior side. So this is what seen in the regular projection. This is how seen in the amphibians. So here, this is the retina. You have the dorsal side, you have the ventral side, you have the anterior side, the posterior side. This is the cartoonic representation of the tectum, showing the part of the brain, showing anterior side and posterior side. So the neuron mostly from the nasal side projected to the posterior side, the temporal side of the retina projected to the anterior side. So this is seen in the normal frog. Okay. So this is how this is more simplistic way, but this is what is shown in other way. So here you can see that this is the retina and this is the tectum of the brain, and this is the retina of the frog in the eye. So you have the dorsal side, you have the ventral side, you have the nasal side, you have the temporal side. Okay. So mostly the neuron from temporal side, they project into the anterior side of the tectum. Okay. The temporal side project into the anterior side of the tectum. Similarly, the neuron from nasal side project into the posterior side of the tectum. Nasal side project to the posterior side of the tectum. Okay, so this is shown in other way. But when this frog encounter a kind of prey, the fly, because it extends its tongue, then it will rotates its eye. So then you have different arrangement, and suddenly the projection of the neuron, which you are seeing here, like the nasal side projecting to the posterior side, the temporal, the nasal neuron from the nasal side will project on the posterior side of the tectum, and the temporal side will project to the anterior side of the tectum. So when it's kind of rotates its eye, then the projection will be changed. So that is what is shown here. So here you can see that nasal will project into the posterior and temporal will project into anterior. But when kind of it's rotated its eye, you can see that the nasal, you have the orientation of this, the orientation of the tectum will change. It was oriented in that direction. Now you can see that oriented in this direction. So the temporal will project in the project in the anterior side, and the nasal will project in the posterior side. The orientation will be, the projection will be same, but the orientation of the tectum will be changed. So that is what is shown in the when they show the rotation of the eye. So depending so there are a lot of things I said about axon guidance or axon projection. It's not only the chemicals, the Hox genes, and the contact diffusible molecules. You require the contact, and also you require here showing that how the change in posture can also regulate the projection of the neuron to different compartment of the brain. Okay. So here and depends on this thing depends on the signal. So here the regular kind of thing that you are seeing here, this is the regular arrangement of this or projection of the neuron, but and this all depends on the kind of signal. So here in the retina you have afferent signal. Okay, sorry, in the retina you have the afferent receptor, and in the tectum you have the afferent signal. Okay, so in the posterior side you have more afferent signal. You can see that more magenta color or the pink color, and in the anterior side you have less afferent signal. It is kind of white. Okay, so the nasal, again in the retina you have more signal in the temporal, more receptor, afferent receptor in the temporal side, and you have less afferent receptor in the nasal side. So the nasal neuron is projected to the afferent, where you have projected to the posterior side, because you have more signal. So this kind of like you have less receptor, more signal, more receptor, less signal. So you see this kind of kind of things, this kind of projection in, of the axon to different compartment, and it all depend on the type of signal they express. It depends on orientation of the your structure. Like here in the case of eye, you can see that when it orients its eye, it is so different arrangement of the tectum. So here, kind of reversed oriented here. So the thing is that the axon guidance is a multi factorial process. It requires several things, like it requires cell to cell contact, or diffusible molecules like SIMA3A or afferin A. It requires sometimes the signal present in the target tissue, like if you have some kind of chemoattractant present in the muscle, even if you displace its neuron to different compartment, it still show projection to the muscle, because you have this chemoattractant which can guide the axon to reach it there. So a lot of things, it depends on the interaction with the Hox genes, or sometimes the posture change can also regulate the axon guidance, like at least in case of retinotactile projection in the amphibian eye, mostly in case of frog. So if you have any question, I can take your questions. So this is all about the axon guidance. Again, the questions will come in the exam, the analytical type, not kind of you don't have to remember.